Welcome to our YouTube class on bright field microscope. A bright field microscope is the most common type of light microscope, widely used in biological and medical research to observe transparent and stained specimens. Here's a detailed explanation of its components. Working principle, advantages and limitations. Components of a bright field microscope. Eyepiece or ocular lens, the lens at the top through which you look. Usually has a magnification of 10x or 15x. Objective lenses. Found on the rotating nosepiece, typically 3 to 5 lenses. Magnifications vary, commonly 4x, 10x, 40x, and 100x. The 100x lens is typically an oil immersion lens for higher resolution. Stage, a flat platform where the specimen slide is placed. Equipped with stage clips to hold the slide in place. Illuminator or light source. Located below the stage, it provides light that passes through the specimen. Modern microscopes use an LED or halogen bulb. Condenser. Positioned beneath the stage, it focuses the light onto the specimen. Includes an iris diaphragm to control the intensity and contrast of light. Coarse and fine focus knobs. Coarse focus knob is used for initial focusing, especially with low magnification lenses. Fine focus knob is for precise focusing, particularly at high magnifications. Arm. Connects the body tube to the base and is used to carry the microscope. Base. The bottom part that supports the microscope and houses the light source. Body tube. The part connecting the eyepiece to the objective lenses, ensuring proper alignment for magnification. Working. The microscope has a built-in light source, typically an LED or halogen lamp, located beneath the stage. This light is essential for illuminating the specimen. The light from the source is focused onto the specimen by the condenser lens. The condenser ensures that the light is concentrated and evenly distributed across the specimen, providing a bright background. As the light passes through the specimen, different parts of the specimen may absorb, transmit, or scatter the light depending on their density, thickness, and staining. The variation in light absorption and scattering creates contrast between different parts of the specimen. Stained specimens usually exhibit better contrast because the dyes increase the light absorption of certain structures, making them more visible against the bright background. After passing through the specimen, the light enters the objective lens. The objective lens, which is close to the specimen, magnifies the image. Different objective lenses with varying magnifications such as 4x, 10x, 40x, 100x can be used depending on the level of detail required. The light then travels through the body tube to the eyepiece, where the image is further magnified. The eyepiece typically provides an additional 10x or 15x magnification. The combination of the objective lens and eyepiece creates a magnified, inverted image of the specimen. This image is what you see when looking through the eyepiece. The background of the image appears bright, while the specimen, depending on its properties and any applied stains, appears darker or colored. The contrast between the bright background and the darker specimen makes it possible to observe fine details. Advantages: ease of use, cost-effective, suitable for a broad range of specimens including cells, tissues, and microorganisms, especially when stained. Limitations: low contrast for unstained specimens. Limited resolution. Bright field microscopy provides a flat, two-dimensional image, which may not fully represent the three-dimensional structure of the specimen. Applications: used to observe the microscopic anatomy of tissues and cells. Essential for observing microorganisms like bacteria, fungi, and protozoa. Used in pathology labs to diagnose diseases by examining stained tissue sections and bodily fluids. Thank you for watching. Please like, share, and subscribe to our channel for more informative videos.